Hi, everybody. Welcome to the muscular system. Today, we're going to continue on the review from 21A. So, first part about the muscular system that hopefully you guys remember was covered extensively throughout the semester, probably beginning with tissues, because these are the three different types of muscle tissues that you guys should have talked about. So there's skeletal muscle, which is the muscle that you guys would have talked about throughout this entire system. It's voluntary and found everywhere where you have conscious control over your muscles, so like all over your body. Cardiac is found only on the heart. It is striated, so it has the actin and myosin overlapping in these forms that you guys can see. They also have intercalated discs, which are junctions, if you guys remember, between, and we'll talk about these more this semester, between the cells of the heart so that they beat autorhythmically. And then they're also branched. Last type is smooth muscle. This is going to line basically all of your visceral organs. So, and it's involuntary. So, like lines your digestive system, your reproductive, your urinary, all those types of systems where you don't have to think about the contractions. Muscles have so there's different types, but one of the main types is antagonistic. So, antagonistic means that the muscle groups are going to move the bones that they're attached to in opposite directions, right? That's antagonistic. So while the triceps relaxes, the biceps contracts and is going to allow the angle between the arm to decrease. It's going to help with flexing the arm. The triceps then, when it contracts and the biceps relaxes, is going to extend the arm. Those are antagonistic muscle groups. So if you guys remember, this is the organization. So you've got a whole muscle, which then is going to branch off into fascicles and then fibers or cells. Those fibers are going to contain the myofilaments, and the myofilaments are the myofibrils, and those are going to be the thin and thick filaments that then can be broken down into sarcomeres. So we'll talk about all of these extensively in just a second, but they're all very, very, very important. Here's the structure, if you guys remember, of the muscle. So this is just going to be the muscle cell with the nucleus on the outside. The blue, if you remember, the webbing is going to be called the sarcoplasmic reticulum. The yellow lines in here are going to be called T tubules. Those are also called transverse tubules. The mitochondria are going to be found all over the place because the muscles have to have lots of energy to move. And then if you pull out one myofibril, you're going to be able to start to see the actin and the myosin. Myosin's the thick filament, actin is the thin filament. When you pull the myofibril out on its own, now you can start to see the sarcomere. And again, you guys, the sarcomere by definition is going to be the contractile unit, right? So it's the functional contractile unit of a muscle, which is going to be from Z disc to Z disc. In the middle, you've got the H zone with the M line. The I band is just the actin. And then the A band is the overlapped portion of the thin and the thick filament. Here's the organization again, right? So you've got the H zone, the A band, the I band outside, and then the M line is going to be the middle. So the M line you guys can see is going to be linked with accessory proteins. Those are going to help anchor the myosin in the middle. The H zone has just myosin. The I band has just the actin or the thin filament, and then the A band is going to have both, the overlap between the actin and the myosin. Okay, so here are the proteins that come into play, and they are really important. So the proteins that you guys are going to need to know when we talk about the muscle contractions as review. First protein is myosin. So myosin's called the thick filament. It has a tail and then what we would call here a globular head. The actin is going to be the thin filament. That's the second protein. The actin is going to be the thin rope. The rope, when you basically zoom in on it, you're going to start to see the other two proteins. So the other two proteins are going to be called troponin, which is this gold bead right there, and then tropomyosin, which is that green rope all the way around. So 4 proteins that are really important again, you guys: troponin, tropomyosin, actin, and myosin. There's the myofibril one more time. Okay, so what happens, you guys, in regards to a contraction? I'm going to run through the whole thing with you guys a couple of times, but this is definitely the part that you guys need to know for the review. So when we talk about the myosin, actually, no, let me go back for that. So when we talk about the myosin cross bridges moving the actin filaments, it's really important because that globular head is eventually going to attach to this active site, which in this picture, it looks gold. The active sites are covered as well. But the myosin head attaches to the actin, and then when ATP is present, the myosin heads are going to pivot and basically pull. When they pull the actin towards the midline, and the zone of overlap will increase. So in order for all this to happen, obviously calcium has to be present. You have to have lots of it, and then you also have to have ATP. So I'm going to go through the whole process for you guys, beginning to end, if I can find the best place to do that at. This is pretty far into the contraction cycle. That's into, okay. So let's zoom out for just a second here. This is going to be the excitation, contraction, coupling, and relaxation. This is basically everything you guys need to know from the beginning to the end. When I jump back, this is just everything blown up a little bit more and then into a little bit more depth where you're talking about the hydrolyzing of the ATP, which is still important. I can get you there from here though. Perfect. So let's start here. This, if you guys are tracking, it goes through 3 different parts. The first part is initiation. The second part is the actual contraction. And then the third part is the relaxation. So you've got ABC. So starting with A, so you have initiation of the action potential. Basically what you have here is this blue part is going to be the neuron. The orange part is the muscle. So this is going to be called the neuromuscular junction. Again, if you guys remember back from 21A, the neuron and the muscle don't actually touch each other, which is why you see this like pink space in the middle. It's called the synaptic cleft because they get really, really, really close to each other, but they don't actually touch. So what the neuron is sending out is a neurotransmitter, which is a chemical. Chemicals are going to be secreted out. The way that they are moved is through exocytosis through these vesicles. So the the nervous cell is mostly called a motor neuron. So as seen there, the motor neuron is going to secrete out a neurotransmitter. The most common neurotransmitter we'll talk about is acetylcholine. So we'll call it specifically, the neuron sends out acetylcholine through the synaptic vesicles, which are going to fuse with the presynaptic membrane and have the neurotransmitters secrete across the synaptic cleft. Those little red dots are the acetylcholine. The acetylcholine secretes across the synaptic cleft and then binds to the postsynaptic membrane, which you guys can see is this ridge in here. The postsynaptic membrane is on the side of the muscle. When the neurotransmitter binds to the receptors in the postsynaptic membrane, what happens is an action potential gets fired and it travels down the T tubule. Again, you guys, the T is the transverse. So the action potential, if you guys are tracking, a chemical signal, you guys got converted into an electrical signal in the form of an action potential, and it's going to travel down the T tubule. The T tubule then propagates that signal out to the sarcoplasmic reticulum, and the sarcoplasmic reticulum is then going to release the calcium that it holds. It does so by attaching to the ryanodine receptor channel, and then it's going to allow more calcium to come out. When the calcium comes out through those receptor channels, it initiates the action potential. So what happens when the calcium comes out is the calcium is then going to interact with the troponin. The troponin then is going to, we're here at step 5. Calcium binds to the troponin. The troponin is going to release its grip on the tropomyosin, and the tropomyosin is going to roll off of the binding sites. When the tropomyosin rolls off the binding sites or the active sites are now exposed, and the myosin head is now going to bind to the active site. So you guys can see the myosin heads are going to bind. When the myosin head is bound to the active site, when the presence of ATP is there, right? So when ATP is present, then it's going to hydrolyze and it's going to power stroke over, right? Power stroke, basically pull over, and it's going to pull the actin towards the midline, which means that the sarcomere itself is getting shorter in length. So the actin filament slides toward the center of the sarcomere, and the contraction has occurred at that point. To release all of this, you have to have so they didn't tell you. Okay, so if you jump back, right, acetylcholine was the neurotransmitter that was bound in the beginning. So what you're actually going to need is you're going to need an acetylcholine esterase. So it's ACHE, which is an enzyme that's going to release the acetylcholine from the binding site, and then it's going to stop the signal being sent down the T tubule, stop the signal getting sent to the sarcoplasmic reticulum, which means that now instead of releasing the SR, you're also going to add in an ATPase that's going to help pump the calcium that was just used back up into its storage site of the sarcoplasmic reticulum. So you're going to restore the calcium back in the SR, which now means that the binding sites are open again. The troponin is going to roll over and tropomyosin is going to roll over and cover the binding sites. The troponin is going to grab on and hold tropomyosin in place, which means the cross bridges have now been broken again with the use of ATP, and now the muscle can relax and regain its original length. So that is, in short, what you guys need to know for the entire section. Obviously, there's more details to it than that, but that's basically what you need to know all the way from the beginning to the end. If I jump back into these, you can start to see the troponin and the tropomyosin for the relaxed versus the contracted state. So you'll see that the active site is on the actin bead, or the molecule, if you will. Then the myosin head will bind, obviously with the presence of ATP. When you hydrolyze the ATP into an ADP plus an inorganic phosphate, then you were going to be able to use that energy and power stroke over or have the contraction occur. That's the whole process. This is the T tubule to the SR, which is going to release the calcium. And we covered that. Okay, I'm going to move forward, but if you guys have any questions on any of these steps, please, please, please let me know, because this is very, very, very important. Okay, so moving into the timing of the EC coupling. So all of this is going to happen in a very, very, very short amount of time. Your muscles pretty much work instantaneously. So when we say instantaneously, it's like milliseconds. So there's different periods that you're going to talk about and go through. So a single contraction relaxation cycle is what we would call a muscle twitch. There is a delay between the muscle action potential and the beginning of the tension. That's actually going to be called or termed the latent period. So the latent period you guys are going to see is right here, where the stimulus has occurred, but nothing has actually happened in the muscle. So if you guys review back, and I'll jump into the nervous section in just a little bit. If you review back to the nervous section, basically what happened is, hopefully you guys remember, so there's different types of neurons. There's sensory, and then there's motor. So the sensory neuron, you guys, carries a signal into the CNS. So we'll start on that side. So there was a stimulus. The sensory neuron picked up the stimulus, carried the signal into the central nervous system. The central nervous system interpreted the signal and sent a signal out through the motor neuron. The motor neuron then secreted the acetylcholine, or the neurotransmitter. It secreted across the synaptic cleft. It binds to the postsynaptic membrane. It then sends the chemical signal and converts it into an electrical signal. Then the electrical signal travels down the T tubule. The T tubule then propagates it to the sarcoplasmic reticulum. The sarcoplasmic reticulum then releases the calcium. The calcium then binds to the troponin. The troponin releases the tropomyosin. The tropomyosin moves off the binding sites. Then actin and myosin are now bound. That's all the latent period, you guys. After which point now you can have the contraction occur. So once the actin and myosin are bound with the presence of ATP, now you're ready for the contraction. But everything that I just went through, you guys, happens very, very, very quickly, right? You're talking just a couple of milliseconds. So that's going to be termed the latent period. The contraction phase is going to be represented by the tension increasing in the cross bridges, right? The cross bridge between the actin and myosin. And then the relaxation phase here at the end is going to be the tension decreasing when you're going to regain resting membrane potential and the full muscle length. Let's see. Okay, so skeletal muscles require a constant supply of ATP. So ATP is going to be used to have the contraction occur as well as to have the relaxation occur. So if you need a lot of ATP, where are you getting it from? So muscle fibers use ATP constantly. The amount of ATP in a muscle fiber is only sufficient for about 8 twitches or approximately 5 seconds. So ATP has to be accessed in different ways. So what this chart is showing you guys is that phosphocreatine can sustain only about 10 seconds of maximal exercise. So ATP production is very high, muscle endurance is very low. Can't use it for very long. Anaerobic metabolism, you're going to get a little bit more ATP production out of there, almost 3 ATP, but ATP, you're only going to be able to use this for maximally a minute of exercise. So it went up just a little bit, but you still can't do very much with it. So you're talking about without oxygen. Um, every time you guys exercise, the beginning of your exercise, I'm not sure if you realize this, it is anaerobic respiration because it takes a little while for your oxygen supply to increase so that you're actually transporting the oxygen to the muscle cells in your body to the point where you're now utilizing them for your breakdown. Aerobic metabolism is really where you want to stay. So, aerobic metabolism, although ATP production goes way down, you can sustain this for lots and lots and lots of time. So, aerobic metabolism can support exercise for hours. Think about like long distance runners, right? They can literally run for hours on end with plenty of energy to do so. So, aerobic metabolism is obviously where you guys are going to spend the majority of your time at. Phosphocreatine, right? So here's the the resting muscle stores energy for ATP and high energy bonds of phosphocreatine. So working muscles then use that stored energy. So ATP comes from metabolism plus creatine. You're going to use an enzyme called creatine kinase and then make phosphocreatine in the body. Working muscle, though, right? Phosphocreatine plus ADP, creatine kinase, and then you're going to now break it down to creatine plus the ATP. Then you're going to use that ATP for all the contractions that you're doing. This is intensity. Um, so exercise, mild exercise, moderate exercise, and then heavy exercise versus the intensity on the side and the percentage of energy expenditure. So obviously heavy exercise, you're expending a lot of glycogen in the body. You're breaking down some fats and a little bit of glucose. Mild exercise, basically you're breaking down a lot more fatty acids and then depending on the duration of time, right? If you're working out for longer periods of time, obviously you can't sustain heavy exercise. So you're just down to these two categories of mild and moderate, and the majority of what you're breaking down is fatty acids. Fun fact, the best time for you guys to work out is going to be in the morning because it's what your body's going to be working in what you would call a fasted state, which means that you haven't had food probably in the last 6 hours, which means that your body's already gone through all of its, glucose and such, and now you're ready to break down fat. Because there's nothing else in your body that your body can utilize for energy. So in the morning is the best time to work out if you're trying to break down fat. This is the anaerobic versus aerobic again. This is the overview of muscle metabolism. So what this is showing you is where your body can get the energy from, right? So glucose you're going to store in the intestine, it can be absorbed from the intestine, put it in the bloodstream. Glucose is not going to be stored as glucose, though you're going to convert it into glycogen because glucose is so easily metabolized, you can't really store up a lot of it. So, glycogen, right? Liver is going to store glycogen. Then you would convert it back to glucose and put it in your blood. Or you can convert back lactate to pyruvate to glucose and then put it in the blood. From your fat, you can break down glycerol and fatty acids from the triglycerides and then put the fatty acids into the blood and then into the muscle tissues. And then they could eventually jump into the metabolic cycle and create ATP for you. Or your lungs, right? Gas exchange with the lungs, oxygen is going to come in through the blood and it's going to help power that cycle of creating ATP. So here's your energy substrate use during exercise. So percent intensity is going to be X and Y is going to be percent substrate used in muscles. So fat versus CHO. um, which is your glucose. So obviously, it's going to go up for the glucose and then down for the fat in regards to percent intensity. So percent maximum, right, is going to go down this way, and then glucose goes up. So what it's telling you, at low intensity exercise, your muscles get more energy from the fat than they do from the glucose. And then during high intensity exercise, your muscles are going to glucose becomes the main energy source because you've depleted all the fats. This is your, okay, so this is lactate threshold, and a calculation that you can do to see um, how many like calories per day you really need. So it ranges from 10 on elderly and sedentary, 80 for different types of athletes. We're not going to do this calculation because that's a little bit too much in depth for us. So I would say stick to what we've already talked about um, for this portion of the review. Oxygen consumption obviously is very important. You have to supply your cells with plenty of oxygen so that you can continue to make energy to power the cells for the contraction. Muscle fatigue is obviously an issue. Your muscles are going to literally fatigue when they have no more energy to continue contracting or relaxing. This is where you could also include talking about rigor mortis. So, rigor mortis obviously is Latin. Rigor is stiffness, mortis is after death. So, after death, a body is going to stop producing more energy or ATP, and its supplies of ATP are going to be just completely exhausted. So the muscles are going to enter a period of what we would call rigor mortis, which is stiffness. So the stiffness is due to the cross bridges of the actin and myosin that have become bound to each other. The contraction has occurred, but it's immovable because you do need ATP to release the contraction. When there's no more ATP present, basically what happens is the cross bridges are stuck in the contracted state. Rigor mortis can last somewhere between 24 and 48 hours until the enzymes within the cells begin to break down due to the lack of anything else in the body. At which point, then everything would kind of go limp. But fatigue can happen. Fast twitch and slow twitch muscles. This is just different types. Not super important for our review, but you've got slow oxidative and fast. So slow twitch oxidative and fast twitch glycolytic, which just has to do with the different types of myoglobin, hemoglobin, color. They're different because they work for different things. So fast twitch muscles obviously fatigue faster, but they work faster. Slow twitch muscles fatigue slower. So like fast twitch muscles would be found in your eyes, or in the areas that sprinters would use, whereas slow twitch muscles, you'd find them in like your torso or your legs, right? Where you could like walk for miles and miles and miles and miles, right? People literally walk cross country and they're fine. Here's the review for you guys on the characteristics of each muscle type and where they're found. Here is a length versus tension relationship on muscles. So basically, the more tension you have, the more contraction force you're going to be able to generate. That's probably all we need to say about that. Summation of contractions. So this just has to do with the fact that you can build on contractions. So this is single twitch. Muscle relaxes completely in between the stimuli or the twitch. Summation is now all of a sudden, and it's going to have a stimulus that's closer together. So you've had a contraction and a relaxation. A contraction, you began the relaxation, but then you had another stimulus and another contraction. So what happened is the tension of the summed twitches was higher than the individual ones. If you go down to the next one, right, the summation leads to tetanus. There's a video I can show you guys on this. I'll probably post it for you guys on Canvas, but basically what's happening here is the contractions are happening so close to each other that the muscle is never having enough time to relax completely. So you're building on tension until you hit maximal tetanus. Eventually what's going to happen is you're going to have maximal tetanus that we would also call complete tetanus, at which point there is no relaxation happening at all between the muscle and you just see complete contraction at this point. After a while, obviously fatigue will take place and then the muscles will relax. This is motor units. So motor units are going to be one motor unit and then all of the muscle fibers that they innervate. So muscles have many different motor units and many different types just because there's tons and tons of neurons that have to innervate them. Isotonic contractions versus isometric. Basically, isotonic are going to be muscles contracting, shortening in length, and creating enough force to move the load. So it's movable contractions versus isometric, which is a muscle that's contracting, but it doesn't shorten. So perfect example of isotonic contractions would be like bicep curls. Isometric would be like planks that you would do. And it's that like ab workout where you're basically just holding yourself up with your forearms. You're not actually moving any load anywhere. You're just contracting the muscles. This is the series of elastic elements in the muscle. Don't worry about this for the review. Load versus velocity isn't very important either. And then the duration of the twitch. So all you guys need to know about this, obviously, is that skeletal happen fast, cardiac happen fast, and smooth take a long time for the contractions to occur. All right, that should cover it for the review. Again, you guys, the most important part of this review is going to be the very beginning where we went through the excitation contraction coupling, which is this slide here. If you guys have any questions on that, please email me. Talk to you guys soon. Have a good day.